Moving on, here we are, our third type of organic macromolecule. So we have looked at carbohydrates, we have looked at lipids. Both of those contain both carbon, hydrogen, and oxygen. Those three elements are going to be the main ones. You can tell the difference between carbohydrates from lipids because of the ratio. If you guys remember carbohydrates, monosaccharides, disaccharides, polysaccharides. They will usually be close to, if not in a 1 to 2 to 1 ratio. For every carbon, you have two hydrogens and one oxygen. Your lipids are a little different because they have a lot less or fewer oxygens there. So you have these really long hydrocarbon chains of hydrogen carbons only hooked to that glycerol molecule, and you'll bond those three chains to it. So very few oxygens within that. As we move on to our third type of organic macromolecule, this now is going to be your proteins. So proteins have the elements carbon, hydrogen, and oxygen because in order for something to be organic, it should contain carbon. It usually will have hydrogen and oxygen as well because of the valence electrons and how carbon likes to grab onto things. But now we're also going to see the element nitrogen. So you cannot be a protein without nitrogen within it, and we're actually going to see our functional group or our amino group here. So proteins, the building blocks. So we keep talking about building blocks, what makes up certain molecules. So we know that your carbohydrates, the building blocks are monosaccharides. Your lipids, the building blocks are glycerol and fatty acid chains. Your proteins, your building blocks, your Legos here are called amino acids. So amino acids, remember first year bio, you probably did a little codon wheel. You looked up three base pairs, AUG, CCG, whatever it may be, three base pairs, and it gave you an amino acid. And that's how it codes for in your body from your DNA to your RNA. We looked at codons and anticodons, and we'll get to all that if you're like, holy crap, I have no idea what she's talking about. That's coming. But as we go from proteins into that material, you guys should start to kind of piece some of this stuff together. Amino acids were what you guys saw on that codon chart that your DNA basically codes for. So your DNA codes for your proteins as well as many other things. So once again, proteins are made up of amino acids. There are 20 different amino acids. No, you don't have to memorize them, but you should memorize some things about them. So the sequence of these amino acids creates unlimited number of proteins. Let's think about that. The sequence of these amino acids creates an unlimited number of proteins. So let's say you're sitting at home and when you're a little kid and you have all your Legos sitting in front of you. Each Lego, we can say, is an amino acid. And maybe you only have 20 different Legos. You have some blue ones and some red ones and some green ones and maybe some square ones and rectangular ones. But there are 20 different amino acids. With those 20 different amino acids, I don't care how many of each of them you have. You might have 400 blue ones, 300 green ones. It doesn't matter. But you have 20 different ones. By stringing them together, you can make unlimited structures from them. Think about it this way. There are 26 letters in the alphabet, just like there's 20 different amino acids. How many different words can I make with those 26 letters? Well, I can make an unlimited number of words. I can make one-letter words, two-letter words, three-letter words. I can make super duper, really long, supercalifragilisticexpialidocious words, if you can spell all that. So the way that you arrange those letters and the length of the word itself means I can make unlimited ones. Same thing. If I take amino acid number one, amino acid number one, amino acid number one, amino acid 2, 3, 4, and I start stringing them together, I can make unlimited number of proteins. Your smallest protein is roughly about 50 amino acids in length. Your largest one can be thousands and thousands of them. We're going to talk about different structures of them here in a moment as well. So the sequence of these amino acids creates unlimited number of proteins. There are 20 different amino acids. All right, so I'm going to go to characteristics before I kind of go to functions. So this is something for your flashcards. So if you're sitting close by, pull out flashcards, look up your amino acid structure. I think I asked you guys to identify your amino group and your carboxyl group and your R group. Here you go. So this structure that you guys see right here, this is your basic structure for an amino acid. So it does have certain functional groups to it. This end of it, circled in green, is called your amino group. It's an NH2 group. Okay, so this is going to have more of a positive ending to it. There's my nitrogen, my element nitrogen. On the other end, that blue square that you see there is a carboxyl group. It's the same thing we saw on the end of our fatty acid chains. Carbon, double bonded to oxygen, bonded to OH, and then it's also bonded to this central carbon. This carboxyl group has more of a negative charge to it. Think about it, because we're going to be putting these proteins into the gel electrophoresis, and we're going to kind of be separating them out based on some of these structures that we see here. Both your carboxyl and your amino group are centered around this particular carbon. So you have a central carbon, carboxyl group, amino group. You will always have a hydrogen, and then whatever else you have up here is known as your R group. This R group is what makes each of the 20 different amino acids different. So we said that R group is a variable. There are 20 different endings that could go here depending upon that particular amino acid that we're looking at. It might just be hydrogen there. It might be a carbon with a bunch of hydrogens around it. It might be a phosphorus in there. It might be other elements in there. But it is one of 20 different endings that gives that amino acid its characteristics. So we have a lot of different functions here, unlike your carbs, which are just like your energy storage and structural, and your lipids are secondary energy storage and protection, waterproofing, that sort of thing. We have a lot of biological functions in proteins, and you guys will see this over and over and over again, and we'll do different things on it in here because it plays such a large role within the human body. So your first function that we see here is that your proteins can be structural. So proteins that are structural could be collagen within your skin. It is a form of connective tissue. It's right underneath your skin, and basically it keeps your skin supple, so it keeps the wrinkles away. So as you get a little bit older and those proteins begin to break down, what happens, that collagen begins to break down, kind of that netting that you have there begins to break down, and people develop wrinkles as they get older and older. It's because of the protein within their skin. So you may see commercials, Oil-Olay, retinol, all these different commercials that talk about how can put that collagen back in the skin, put the lotion on, and you're helping to basically keep that protein nice and taut and tight in that netting underneath your skin. It can be regulatory. So we said that some of your hormones are actually lipid-based, your cholesterols and your estrogen and your testosterone. Some of them are actually going to be protein-based as well. So insulin makes within your pancreas. It helps to regulate your blood sugars. Those people who are diabetic are not able to make insulin, and they have to get it from another source. Oxytocin is another one. We'll talk about that a little bit later on when we get to some of our human stuff. Contractile, proteins that are contractile, actin and myosin, your muscles, muscle contractions. So when we get to histology and we talk about different types of muscles and we look at the banding and actin myosin bands within them, it's actually protein bands that you're able to see. Transport, you guys will be doing a lab on myoglobin. You guys will be doing a lab on hemoglobin. These are two types of hemiproteins, which means that they are iron components within your blood. Your myoglobin is going to transport oxygen more towards your tissues. Your hemoglobin is going to transport oxygen within your blood in order to get it to those tissues. So we'll be seeing them a little bit more in your protein lab if we haven't done it yet. Storage, egg whites. We know that egg whites are a great source of protein there. Basically, that's, as that chicken and other organisms are kind of forming within that substance, that's what they're kind of feeding on. Also, seeds as a place to store protein as it's produced from different organisms, such as plants. We find it within the membrane. So when we went to phospholipids, we said, oh, you know what? Cell membranes. If you remember the word phospholipid bilayer, we have, fatty acid tails, and they're sticking together and they're hiding away from the inside and outside the cell. And your phosphate heads are facing out of the cell and into the cell where there's mostly water. And I said, well, we also refer to it as a fluid mosaic model. And the reason is because that cell membrane is not only phospholipids, it's got proteins in it, it's got cholesterol in it, it's got some carbohydrate little receptors in it. It's all these little pieces that are kind of joined together, kind of like mosaic art, which is, all different colors and shapes and things that are kind of thrown together to make that particular artwork. That's what we see with our phospholipid bilayer. So it also makes up the cell membrane. We have protein receptors within it. It helps to make channels within the cell membrane so that larger molecules can move back and forth. Proteins can also be toxins within certain organisms. Bee sting, venom of snakes, jellyfish stings, botulism produced by bacteria. These are all toxins that are protein-based. So the organism is able to produce it, and it is actually toxic to other organisms. The next one you're going to see is START, enzymes. So enzymes we'll do a whole section with. more towards the second half of the year. But enzymes basically are a catalyst. What that means is that they help to speed up reactions without being used up. They can be used over and over and over again. So earlier on, we talked a little bit about being lactose intolerant. And I said, well, if you're lactose intolerant, you are not able to break down the milk sugar lactose into the smaller monosaccharides, glucose and galactose. Remember, lactose is a disaccharide, two sugars joined together. We want to break it apart. We do this all the time in our body. We have an enzyme lactase in order to break down lactose because enzymes are very specific. And I said, well, if you're lactose intolerant, it doesn't mean that you can't break down milk sugar. You can still do it. It's just that enzyme lactase is going to help speed up the reaction millions of times faster. If you don't have the enzyme, you have a little mutation in your DNA. It just takes a lot longer for your body to do that. And a lot of times that breaking down process is going to tend to upset your stomach along the way. We'll talk about how enzymes are able to be used over and over and over again unless you denature them. But you can denature them with heat. You can denature them with different pHs. So within your body, let's say your mouth pH of 7, your stomach is a pH of 2, your kidneys are a pH of 9, and you have different enzymes that will work in each of those areas. If I take the amylase within my mouth, which lives in a pH of 7, and I stick it in my stomach, it's not going to work because that pH is a 2, and it's going to denature or destroy that particular enzyme. And my body has to go through, find the code in my DNA, and make more of that enzyme if I need it. And we're able to do that. So where I say it does kind of go through and get destroyed, we can go through and make more if the correct amino acids are within our body. If we're not eating a balanced diet and we don't have the Lego pieces to put together to make that particular Lego structure, it's not going to happen. You have to eat the balanced diet. You have to get the correct amino acids to put them together through dehydration synthesis in order to go through and make that protein. So we said that they're very specific. So they'll usually only work on one thing. So sucrase is the enzyme, ends in ASE, breaks down sucrose. Lactase is the enzyme that breaks down lactose. You have pepsin, it's going to help to break down proteins within your stomach as well. So they will control the rate of reactions. They can be used over and over and over again. We refer to them as catalysts. There are other things that are catalysts as well. It doesn't always have to be protein based. That will help to control the rate of reactions. And when we get to enzymes, we'll talk about them a little bit more. So levels of organization. So there are 4 levels of organization depending upon the size of that particular protein. Remember I said your smallest protein is about 50 amino acids. Well, that's going to be kind of your primary structure. So our levels of organization, that primary structure is just a chain of amino acids. So we used to do this really good visual within class. Can't really do it now because of COVID. But when we had this classroom, 28 students, you guys are sitting next to each other at desk, we would say, okay, each student is an amino acid. So you're sitting there and you're sitting next to someone's behind you. You are an amino acid. And there are 20 different amino acids within the room. Even if we have 28 students, I have repeats someplace along the line. And I would say, okay, amino acid number 1, join hands with amino acid number 2. We just join 2 amino acids. And when we do that and we join those 2 amino acids together, we create a dipeptide. Di meaning 2. Peptide is 2 amino acids joined together. How do we do that? Same way we did carbohydrates, the same way that we did lipids. We are going through dehydration synthesis. I need to remove water so that I can bond together these 2 amino acids and create a bond through them. That bond that we just created is called a peptide bond. So a peptide bond joins 2 amino acids. Every time I join another amino acid on, I take out another water, I make another peptide bond. 2 amino acids joined together is a dipeptide. 3 or more, just like sugars, mono, di, poly, I have a polypeptide. So 3 or more amino acids joined together is a polypeptide. Another name for polypeptide is protein. So a polypeptide is proteins. So your primary structure is just a chain of you guys. So it's like if I said, okay, my whole first row of students, all 5 of you guys are all joining hands, amino acid, amino acid, amino acid, amino acid, amino acid. I take water out, every place that you're joining hands, I have a peptide bond, every place that you're joining hands, you are a primary structure. You are a chain of amino acids. My secondary structure is called either a beta pleated sheet or an alpha helix. So once we had everybody within the room, all 28 of you guys kind of connecting hands, I'd say, okay, let's go for a little walk. and without breaking bonds, without breaking the hands that you guys were holding between, because every place you were holding hands was a peptide bond, we kind of take a little walk around the circle. And you guys would be this huge chain walking around, holding those hands, which were your peptide bonds. And as you were walking around the circle, you were that primary structure. Then we'd come back into the room, and you would have to get back into and kind of weave your way through the seats without breaking hands. As you did that, and you were kind of zigzagging back and forth, you were creating a beta pleated sheet. A beta pleated sheet kind of looks like this. So I took this piece of paper and I folded it. So amino acids, so we have a chain of amino acids together, and they're folding, and they're folding, and they're folding. That would be that beta pleated sheet. So it gets a little bit more complex. We also have something called an alpha helix. So here is, I probably shouldn't have picked black if I was going to wear black today, but here is my chain of amino acids. So pretend this is all 28 of you guys holding hands. An alpha helix, what happens there is it gets a little more complex. It begins to spin. I was going to use the phone cord, but nobody really knows what phone cords look like anymore either, because nobody really has cords on their phones within their houses. But it would look something like that. So that would be your alpha helix. I'm taking it and it's just becoming a little bit more complex. So that secondary structure is either going to be that beta pleated sheet where it's folded. You guys are folding each other within the room, within your seats. Or if I said, okay, everybody's still holding hands, kind of spiral around each other. That would be your alpha helix. Tertiary structure. Now we're still sitting there within those seats, within that beta pleated sheet or that alpha helix, and we're going to start creating sulfur cross bonds with our secondary structure. So as you're all there within your seats, I say, okay, you're still holding hands, you're still lined up in your seats. Take a foot and touch someone behind you, next to you, in front of you. So it's kind of like if I take my little beta pleated sheet and now I start connecting them and I start making little bonds across, connecting all these particular sheets. Those are my sulfur cross bonds that we have. That is a tertiary structure, so we're getting a little bit more complex there. These are disulfide bonds that we're going to see joining those beta pleated sheets, or if I had the alpha helix, it would be between all those little spirals there as well. So here's my, once again, if I had all these little disulfide bonds joining each of these little coils all the way down, that would be that tertiary structure. Quaternary, your fourth level structure, 2 or more chains. So if I took this particular class and I joined it with Mr. Spate's class and they were doing the same thing, and I did it with another class of mine, and I had all 4 classes together, and you guys are all holding hands and connecting feet, that'd be a huge globular mess. And that's basically what a quaternary structure is. It's 2 to 4 chains bound together. It is a huge globular protein. And we'll talk about some of these quaternary structures, these giant proteins that we see here. Collagen is one. It's a huge netting structure that begins to break down over time, but it is a globular protein. Hemoglobin is also a globular protein. It is made up of 2 beta sheets, so 2 sheets that look like this, and 2 alpha helixes, so 2 of your strands that look like this. So I put them all together, and once again, it is a globular mess. It is a giant protein. When you guys do your protein lab and you put it into gel electrophoresis and you put it into those little wells and we let it run for 20 minutes, and at the end, you're going to go, it's still in the well. It actually is still pretty much going to be in the well. It is a humongous protein. So remember, these things are moving within the gel based on molecular mass. If it's got a huge mass to it, it's going to take a very long time for it to move, or it's barely going to move. So hemoglobin, once again, globular protein. It is the oxygen carrying component of your blood. So it goes from your tissues, or from your lungs to your heart, goes and drops everything off the tissues, and that's where myoglobin is going to come in and transport that into your tissues. Collagen is tertiary to quaternary depending upon where it is. It's that network-like support that I was just talking about. It's found underneath your skin. As you get older, that connective tissue, that protein begins to break down. It's basically what keeps you young looking. Your skin looks supple, looks nice and tight. Kind of hard to stop it. You put some, all that moisturizer on. Try to put excess proteins into your skin to help with that. It's actually, I'm not going to see this on the test, but it's a whole bunch of helicals twisted into a right-handed coil. So it can be super duper complex, just like your hemoglobin. Okay, so we'll be working with this a little bit within your protein lab. You guys will see a little worksheet that goes along with this as well. We'll also be going through diagrams, and we'll also go through handwritten notes. So kind of like let some of this stuff sink in. Thing to remember is that basic amino acid structure, there are 20 different amino acids, carboxyl group, amino group, around the essential carbon, hydrogen, and then the R group. And that R group is the only thing that changes on that amino acid. So I have 20 different R groups. It's a variable. That's all it is. And when I put those 20 different amino acids together through dehydration synthesis, I can make unlimited number of proteins, either a primary structure, secondary structure, tertiary, or quaternary, depending on what type of protein that we're looking at. Okay, and we'll discuss this a little bit more as we go on. Okay, thanks.